Today we'll see the practical number 4th, that is the qualitative test for proteins. Under qualitative test for proteins, we will see the two tests. First of all, we will see the introductory part regarding the proteins. The name protein, which is derived from the Greek word, and the proteos meaning first was proposed by the Berzelius in 1838. Mulder in 1839 first used the term to refer the complex organic nitrogenous substances found in the cell of the living organisms. They are most abundant intracellular macromolecules and are the building blocks of protoplasm of each cell, and constitute 50% of the dry weight of the protoplasm. The proteins also occurs in a plant cell in the form of stored food. The physical and the chemical properties of the reserve or storage proteins differ from the protoplasmic proteins. The proteins with a catalytic properties, that means enzymes, are largely responsible for phenotypic characters of the cell in a particular environment. Thus, the proteins are essential for the cell structure and function. The constituent elements of the proteins are the carbon, hydrogen, nitrogen, and oxygen. The plant proteins also contain sulfur and phosphorus. The main building blocks of the protein molecules are the amino acids. The backbone of protein is formed of a polypeptide linkage of amino acids, the number of which may vary considerably. The union between the amino acids takes place by peptide linkage. It is a linkage between an amino acid group of one amino acid with the carboxyl group of other amino acid, producing a molecule of water in the reaction. The resulting molecule is termed as a dipeptide. This reaction chain is continued to produce the polypeptides, peptones, proteose, and finally the protein is formed. For carrying out that experiment, the requirements are essential. That is the test tube stand, 0.01% anhydrine, concentrated HNO3, concentrated NaOH, water bath, matchbox, gas, water, etc. The plant material which is required, that is the gram flour. The qualitative test for protein, we will see only the two tests. First, that is the anhydrine test. Take a 2 ml of sample in a test tube, add a 0.5 ml 0.01% anhydrine and 2 drops of anhydrine. Heat it in a boiling water bath. What you observed after completion of certain period, in a test tube there is a development of blue color. That means the blue color appears and it indicates that there is a presence of proteins. While the second test, we can call that is the Hydroprotic test. Take a 2 ml sample plus 1 ml concentrated HNO3. There is a development of white precipitate. When heat it, there is a development of yellow color while cooling it. After cooling, you add a few drops of concentrated NaOH. The yellow color changes to bright orange. It indicates that there is a presence of proteins. Thank you.